So hello, how's everybody doing? Welcome back to this channel. I will be your A and AS level biology teacher. So without any further delay, let's start today's lecture. Today we'll be studying cell structure, and our vault for today is magnification versus resolution, microscopes and its types, and basic cell structures. So let's begin with our first topic, that is magnification versus resolution. Yes. Okay, so magnification is basically how many times bigger the image is from a specimen, right? Sorry. How many times bigger is the image from the specimen? From the specimen, yes. So there's a specific formula to calculate the magnification. Yes. So the formula is I equals A into M, where I is the size of the image, A is the actual size of the specimen, and M is the magnification. Now to remember this formula, I have a trick for you. So let's draw a triangle. Yes. Let's draw two lines. I equals A into M. So if you cover any one of them, so let's say you have to find the image, size of the image. You cover image and you will get A into M. But let's say you have to find the size of the actual specimen. So you cover A and you will find I by M. Right? So to find anything, you just have to cover it up and write the corresponding letters. Yes? That's good. Now we are going to learn about resolution. Yes. So resolution is basically the ability. So the ability to distinguish between two separate points, not any same points. Distinguish between two separate points. Yes. If two separate points cannot be resolved, they are observed, or they are said to be one point. So let me type that down. If two points cannot be resolved, they will be considered as one point. sorry. Considered as 1 point. Yes. So that's it. That's all we have for magnification and resolution. Now let's move on to our actual topic, the second learning objective. So we all know what a cell is, right? But you can't just see a cell with your naked eyes. So to see a cell, you need to use a microscope. Yes. So our next topic will be about microscopes. So microscopes are basically of two types. That is a bad arrow. So yes. So one is the light microscope. And one is the compound microscope. Yes. So sorry, not compound. One is the electron microscope. Got confused, sorry. Electron microscope. So we're gonna first talk about the light microscope. Yes, perfect. So in a light microscope, you shine light through a specimen. So let me write that down. Shine light through a specimen. Light microscopes are the everyday normal microscopes we use in your school. You might have electron microscopes are very expensive, so your school, if it has a microscope, it has to be a light microscope. Yes. So you shine light through a specimen which goes through a lens. Which goes through a lens and produces a magnified image. So it's simple light mechanics. Light goes through lens, it refracts. What's happening? Yeah, it refracts and it produces a magnified image. A combination of lenses used over here. So just one lens will not do the trick. You have to have many lenses. So the specimens which you can watch or observe under the microscope, light microscope, can be living or dead. So specimen can be living. Whoa, yeah, oh, what's happening? Can be living or dead. And by living, what I mostly mean is they can be moving or motile. It's another word for moving. Yes. Yeah, so like for example, if you want to see not a bacteria, that's too small, but let's say if you want to see cells like surface cells like cells of the onion peel, then you can watch it under a light microscope. So light microscopes are of two types. Yes, so light microscopes are of two types. I'm gonna write them down right now. So the first one is a simple microscope. Let's talk about a simple microscope. So a simple microscope is used to see the surface structure of an organism, used to see the surface, or if you want to use a better word, the morphological basically means physical, so physical features or physical structures. Physical features of a specimen Yes, is it clear till now? Please copy down whatever you've done till now. Pause the video. I'm zooming out. Yes, so let's begin with our second type of light microscope, which is the compound microscope. Okay, yeah. So the compound microscope is basically used to see the basic cell structures, not too detailed, just the basic structures. So use to see the basic cell structures. Yes. So that's it for the types of the light microscopes. Now let's talk about some pros and cons and some details about the light microscope. So the preparation of the specimen is very easy in light microscope. So you just take a specimen and put it on a slide and add another one on top, so and put it under the microscope. You have to clean the specimen first, of course, so that's fairly easy. Preparation of specimen is easy. Yes, now let's talk about magnification. How many times it can zoom in to the specimen. So usually the light microscope has a magnification of 2000x. So the image of the specimen is magnified 2000 times its original size. Resolution. Resolution of a light microscope is around 200 nanometers. So if you don't understand why I use nanometers, let me go back to the resolution thing. So resolution is basically so resolution is basically related to distance between 2 points. So I'm just gonna rewrite the full thing, please. Yes, so distance between 2 points. So we're done with the light microscope for now. Now let's move on to our second type of microscope, which is the electron microscope. Yes. So the definition of the electron microscope is basically in its name. So electron microscope uses a beam of electrons I'm writing in short forms, you please write in the long one, to produce magnified images. Yes. Electron microscopes are of two basic types. They are of two types. So the first one is the scanning electron microscope. Yes, so the scanning electron microscope, what it basically does, it is technically in the name, it scans the surface of the specimen. So it shows the image of the surface of a specimen. Not just the surface structures. Or if you want to do it, it shows the image of the surface of any structure in the specimen. Yes. Now the second type of electron microscope is the transmission electron microscope. Yes. So the transmission electron microscope shows internal images. Internal images of any organelles. So that's how we know what the structure of many organelles are like. Because of the transmission electron microscope, it has made our jobs a lot easier. So please do write down whatever we've done till now. I have zoomed out. Pause the video if you think that suits you best. Okay, so now let's talk about the pros and cons and the features of the electron microscope. So sample preparation here is not easy. You have to be very precise and intricate with how you handle the samples. is not easy. Yes. Now, one of the most important points, living or moving organisms or specimen or moving non-living things can also move, or it could be in their nature to move. Living or moving organisms cannot be observed under the electron microscope. Cannot be observed. Yes, because it cannot be observed because, see, in electron microscopes you throw, like, let's say this is the beam projector and you throw a beam of electrons. These are electrons, let's say. So the beam of electron bounces off. It just goes here and bounces off the sample, and then it goes to a receiver where it reads the pattern of the bouncing and makes an image. Now, if the specimen is moving, the electrons will not hit the specimen and the image formation will not be possible. So that's why we cannot observe living or motile organisms under the electron microscope. Yes. Now let's talk about the magnification. So the magnification here is huge, massive. So the magnification goes up to 500,000x. That's how high it can go. And the resolution is also pretty high. It can see objects that are 0.5 nanometers apart. Yes. Now we've talked about resolution a lot, and we have talked about, and we've used the term nanometers in there a lot. So let me explain to you what that term means. So let's say this is a specimen, or let it just be like that. Yeah. So now here comes a wave of light. Let me just draw. Give me a second. Whoa. Yeah, so this is my baseline for the waves I'm gonna draw. So let's say I'm drawing some waves here. Please pardon my drawing abilities, I'm not that good. Yep. So this right here, what I've drawn, this is not good, sorry. Again, pardon my abilities to draw or making anything aesthetic. Yes. So this right here, what we've drawn is the wavelength of light that is hitting the specimen. So this wavelength, violet has the least wavelength of 400 nanometers and red has the most of 700 nanometers. So it will be pretty difficult to see things that are violet in color. The whipped gear, we've seen the color gradient. Whipped gear. Yes. So we've seen this whipped gear. So violet has the least wavelength and red has the most. What we drew here was the frequency actually. Yes. So if anything is smaller than 200 nanometers, it cannot be seen by the light microscope. Let's say the distance between this point and this point is 100 nanometers. So a light microscope cannot see through that, because we've seen above that the resolution of the light microscope is 200 nanometers. Yes. So the distance between the points are 100 nanometers. So we'll have to use the electron microscope to see the object. Yes. Okay. Please copy this down. I'm zooming out. I hope you've written this down. If you've done, then perfect. Now let's move on and start with the actual chapter. So we're going to learn, oh god, yes. So we're going to learn about the structures of the eukaryotic cell and their functions. So let me write it down. And their functions. Yes. So the first thing, we'll work our way from the outside to the inside. Yeah, so the first thing you see in a typical eukaryotic animal cell, let's say. So the first thing you see, of course, is the cell membrane, or in this case the cell surface membrane. Yes. So the cells of every cell in this world, every cell is surrounded by a cell membrane. So cell membrane is a universal organelle. Universal organelle. Yes, so now the cell membrane, it helps in the exchange of substances. Exchange of substances, yes. Now the cell membrane is made up of small lipids known as phospholipids. Made up of phospholipids. Let me draw it for you, what a phospholipid typically looks like. Yes. So this is what a typical phospholipid looks like. So it has two parts, the phospholipid. The first one is its head. The head is made up of phosphate, so it is called the phosphate or the polar or the hydrophilic head. Hydrophilic head, yes. So the head is made up of, in the name, it's given in the name, phos. So the head is made up of phosphate ions. It is written as PO4 3 minus, yes. And the second part being tail. Tail is made up of hydrocarbon, or we can call it the lipid tail, or we can call it the non-polar tail, or we can call it the hydrophobic tail. Hydrophobic means it does not like water, and hydrophilic means it loves water. So there are two so the phospholipids form a bilayer. So it's basically like this is one layer of phospholipids and this is another layer of phospholipids, and inside is the cell. So in the phospholipid bilayer, since the head is hydrophilic, it stays on the outside. So I'm not going to draw the whole thing, just 4 of them for your understanding. So the head is always on the outside of the bilayer, and the hydrophobic tails, they are on the inside. Yes. Yes. So the head is on the outside, and the tails are on the inside, because water is generally on the outside of the cells. So water is outside and inside. Yes. Perfect. Please draw the diagram. I'm going to erase it. This one over here. Yes. So I'm just going to draw the membrane again in a much neater way. So this is the outer membrane, this is the inner one. Please bear with me. And the tails, I'm just going to draw them as lines. There are also a lot of things present on the cell membrane, but we don't have them in our syllabus, so we're not going to talk about it. They are integral proteins, carbohydrates, cholesterol, etc. So they are basically present in a structure like this, in a bilayer. We call this lipid, or we can just call this the phospholipid bilayer. Yes. Okay, so now we're done with the surface membrane. So let's talk about the diameter of the surface membrane or the phospholipid bilayer. So its diameter is around 10 nanometers, so it's very small. Yes. Okay, please copy this down. I'm zooming out. Pause the video if you feel like it. Yes, now let's move on to the nucleus. the second organelle and the last organelle for today. So you might have heard about the nucleus, or let's say you have heard about the nucleus. People call it the brain of the cell. Yes. So the nucleus is a double membrane bound organelle. Yeah. It contains chromatin, histone, and the nucleolus. So let me write it in another line. Contains chromatin, histones, and the nucleolus. Histones are proteins, chromatins are fiber that contain DNA. We'll learn about that later. Yes, it is double membrane bound, so let's draw a diagram of it. On the side, yeah. Oh, oops. So I'm going to draw the double membrane as one. So this is one membrane on top, and this is the other at the bottom. So we're going to draw multiple of these. There are gaps between these membranes, which we'll talk about right now. Yes. So this is basically the nucleus. Inside the nucleus we find the nucleolus. Yes. And sprouting from the nucleolus, from the nucleolus is basically these thread-like structures. They are known as the chromatin fibers or chromatin reticulum. So let me label them. I'm going to write them as chromatin over here. This is the nuclear envelope. And these gaps in between, they are known as the nuclear pores. And the small circle inside the nucleus, it is the nucleolus. Nucleolus. And like the cell has a cytoplasm, the nucleus has the nucleoplasm. Attached to these nucleus is, one second, let me use another color, orange preferably. Yes, small dots, don't write them as dots please. So they are known as histones. Histone proteins, let me write them as that. Histone proteins. You don't need to know what the histone proteins are. You just need to know that they exist. Yes. Okay. Please write this down. I'm zooming out. Now let's talk about the function of the nucleus. They contain chromatin, of course, and they regulate cellular activities, intercellular activities. Intercellular activities. Yes, perfect. Write this down, please. Now let's talk about the nuclear envelope right here. Nuclear envelope regulates what comes in the nucleus. Regulates movement of material in and out of the nucleus. Yes, is it clear? This regulation happens via the nuclear pores. Nuclear pores. Yes, I hope that's clear. Now let's talk about the nucleolus. What's nucleolus? Oh yes, found it. That is a really bad arrow. Yes. Oh, sorry, forgot. Please do write this. Pause the video, zoom in, and write the function of the nuclear envelope. So, nucleolus is a very dense and dark part of the nucleus. Very dense. Arguably the densest part in the cell. So what it does is it synthesizes rRNA. Yes. Please write it down. Oh, my pen didn't work. Sorry. It synthesizes rRNA. Oh, one more thing. It is densely stained. Now, what is rRNA, you may ask? Don't worry, I'm gonna tell it. Yes. So the rRNA is basically, you know what RNA is. So I'm not going to write the full form of RNA, it's ribonucleic acid. So rRNA is basically ribosomal ribonucleic acid. This makes ribosomes during the processes of translation, transcription, and duplication, all of that, yeah. Makes ribosomes. Okay, now that we're done with the nucleolus, we'll move on to the nucleoplasm. This is the last part we have to study about the nucleus. Okay, so It's in the name, the nucleoplasm. It acts similar to the cytoplasm. Let me write it down. Acts similar to the cytoplasm. Yes, it's clear till now. It contains enzymes, contains enzymes that help in metabolic activities. Activities. Clear till now? Perfect. It acts like a matrix as well. The matrix, like a cytoplasm acts as a matrix to the cell. The nucleoplasm acts as a matrix to the nucleus. So I think that's about it. Oh no, we have one more thing, the chromatin. The chromatin is nothing, it is made up of DNA. Made up of DNA. DNA is nothing, DNA is made up of genes. So that's about it. So let's revise what we've done, and I'm going to leave you to do your work. Yes. So we first started with magnification. So magnification is basically how many times bigger is the image from the specimen. The formula is size of the image is equals to actual size into magnification. We can use this triangle by covering one. We can find the formula to find the value. Resolution is the ability to distinguish between two separate points. If 2 points cannot be resolved, they are considered to be as 1 point. It is related to the distance between 2 points. Yes. Microscopes are of two types: light microscopes and electron microscopes. Light microscopes uses a ray of light which shines through the specimen and goes through a lens to form a magnified image. Yes. A specimen can be living or dead when using a light microscope. So the light microscopes are of two types: simple and compound. Simple is used to see the surface physical features, and compound is used to see the basic cell structures. Preparation of specimen is easy. Magnification goes up to 2000x and the resolution goes up to 200 nanometers. This means you can see objects that are maximum 200 nanometers apart from each other. Yes. Electron microscope uses a beam of electron to produce magnified image. There are two types: scanning and transmission. Scanning shows the surface of any structure in the specimen, and the transmission shows internal images. Sample preparation is not easy, it's very intricate. Living and moving organisms cannot be observed. They have to be dead. The specimen has to be dead. Magnification should be, or it can go up to 500,000x, and the resolution is 0.5 nanometers, which means you can see objects that are maximum 0.5 nanometers apart from each other. Yes, so we learned about the wavelength. Violet has 400 nanometers of wavelength, red has 700 nanometers. So all of these colors can be seen through a light microscope. Anything smaller is not visible. So now we're going to talk about basic structures of a eukaryotic cell. Cell surface membrane. It is a universal organism found in all cells. It helps in exchange of substances. It is made up of phospholipids. The diameter is 10 nanometers of the phospholipid bilayer. Let me put an arrow to it. Yes, a phospholipid bilayer. A phospholipid is arranged in a bilayer whose nanometer is approximately 10 nanometers. There are two parts to a phospholipid: the phosphate head or the polar head, or the hydrophilic head. Hydrophilic means scared of water. Let me write that down as well. Hydrophilic means hydrophilic means loves water. Loves water. Yes. Okay. It is made up of phosphate ions. Yes. Now the second part to the phospholipid is the hydrocarbon tail, or lipid, or non-polar, or the hydrophobic tail. So what we mean by non-polar and polar? Non-polar and polar basically means whether it is charged or not. Let me write it down. Polar slash non-polar is to see whether it is charged or not, whether it contains any charge or not. Yes. Okay, perfect. It is hydrophobic, which means it is scared of water. Yes. It's scared of water, yes. I try my best. They are arranged in a bilayer because the water is in front and possibly at the back. That's why the head is positioned in this way in a bilayer. Let me add one more thing. The first layer is known as the first leaflet. And the second one is known as the second leaflet. Yes, perfect. Now we talked about our second and last organelle for today, the nucleus. It is considered to be the brain of the cell. It is a double membrane bound organelle, as we can see in this diagram, and it contains chromatin, histones, nucleolus, and regulates intercellular activities. So the chromatin is made up of DNA, and the DNA is made up of genes. The nuclear envelope, let me underline it. Wrong color. Nuclear envelope, it regulates movement of material inside, in and out of the nucleus, like the cell membrane does, to the cell. Let me just write this. Cell membrane is similar, not the same, is similar to the nuclear membrane. Cell membrane does not have pores in it. And the nucleoplasm, as we can see, the nucleoplasm, it acts similar to the cytoplasm. It contains enzymes that help in metabolic activities, and it acts like a matrix. So we can write that the nucleoplasm acts similar to the cytoplasm. Correct? Yes. Okay. So the nucleolus is a very dense and dark and highly densely stained structure. It synthesizes rRNA. rRNA is nothing but ribosomal RNA. Ribosomal RNA is apart or it helps make ribosomes. So that is it for today. I hope you understood everything. I'll try my best to upload or put the link to these notes in the description. I hope you understand everything very well. If there's any questions, any queries you have, please write them down in the comments. I'll try my best to answer every one of them. Okay, so that's it. I'll see you later. So till then, ta-ra, bye bye.